Welcome to DRX Learning Hub. Today, we explore key analysis techniques in pharmaceuticals. Pharmaceutical analysis identifies and quantifies drugs and chemicals. It helps determine both the identity and the amount present. Let's break them down. Chemical methods are traditional techniques based on reactions. Analytical techniques divide into two categories: chemical methods and instrumental methods. They include qualitative and quantitative analysis. Qualitative analysis identifies chemical components. For example, it detects ions or impurities. Quantitative analysis determines amounts in a sample. It includes gravimetric and volumetric analysis. In gravimetric analysis, the analyte becomes an insoluble compound, which we filter, dry, and weigh. An example is estimating sulfate as barium sulfate. Volumetric analysis, or titration, involves a known solution reacting with the analyte. The reaction's endpoint is indicated by a color change. A classic example is acid-base titration. Now let's move to instrumental methods. These advanced techniques are faster and more precise. They detect even trace amounts. Instrumental methods rely on physical properties like absorbance or conductivity. Spectroscopic methods measure a substance's interaction with light. Examples include UV visible spectroscopy for absorbance and infrared (IR) spectroscopy for identifying functional groups. Next, chromatographic methods separate and purify compounds. Techniques include thin layer chromatography (TLC) and high-performance liquid chromatography (HPLC). Electrochemical methods measure electrical properties. Potentiometry measures voltage change while conductometry assesses conductivity. Thermal methods study substances with temperature changes. Techniques like thermogravimetric analysis (TG) determine melting points and stability. To summarize, pharmaceutical analysis utilizes chemical methods, gravimetric and volumetric. To instrumental methods, spectroscopic, chromatographic, electrochemical and thermal techniques. Each method serves unique advantages based on the sample type and required accuracy. In conclusion, understanding these techniques ensures drug safety, purity, and effectiveness. Thank you for joining us. If you found this video helpful, like and subscribe to DRX Learning Hub. Stay tuned for our next topic: limit tests in pharmaceutical analysis.